Today, grade 11s, we are going to be finishing off kind of our notes about the digestive system, and then next class you're going to do a virtual dissection. Woohoo! I know it's not quite the same as your actual pig dissection, which I'm sure some of you are looking forward to, but we're going to do it a little bit differently. We'll just do it virtually this year. So today we're just going to finish off looking at the accessory organs. So the accessory organs that we talked about are the pancreas, the liver, and the gallbladder. So they're called accessory organs because they're not directly on the path of the digestive system, but they definitely help move things along. So we'll look at the pancreas first. So the pancreas produced the pancreatic juice, which contains sodium bicarbonate to neutralize the stomach acid, also known as chyme, when it was entering the duodenum. And it released the 3 pancreatic digestive enzymes to break down food, which were pancreatic amylase, trypsin, and lipase. So the pancreas was in charge of producing the sodium bicarbonate, which neutralized the chyme so that it didn't destroy our small intestine. And it also brought those 3 really important pancreatic digestive enzymes. So to be able to continue on with breaking down our starches, our proteins, and our lipids. So that is one of the accessory organs. The next one we're going to look at is the liver. So fun fact, the liver is the largest gland in the body. If you're ever on Jeopardy and that comes up, there you go. So the liver acts as a gatekeeper for substances in the blood. As blood flows into the liver, it removes poisonous substances and detoxifies them. So the liver is really important in maintaining homeostasis of our blood in our body, especially looking at glucose. We're just going to talk about that briefly here. So the liver helps stabilize blood glucose levels. So when insulin is present in any excess, When insulin is present, any excess glucose is removed and stored in the liver. When there's no insulin present, the liver breaks down that stored glucose into the bloodstream again. So just note that insulin is the hormone that allows your body to use glucose. So if there's extra insulin in the body, that means that there isn't any glucose to break down, so it's all being broken down. So it needs to be extra can be removed and stored in the liver, and then insulin is stimulated by the release of glucose. So when more glucose is detected, or there is a lack of glucose detected, the liver can break down that stored glucose into the bloodstream again, so then we can bring the insulin levels back up. So again, insulin is released when glucose is detected. So it works, that's kind of a negative feedback loop in terms of the insulin and glucose levels being very closely monitored. And so when there is too much insulin, it needs to be broken down. We have to save glucose so that we don't run out of glucose in our body. So we store some, and then when we have too little glucose, glucose is released from the liver, and then our insulin levels go back up as well. So that's really important in maintaining our homeostasis of our blood glucose levels. So the liver also produces bile, which is stored in the gallbladder, and bile contains the bile salts, which emulsify the fats. So the liver is in charge of monitoring both our glucose levels as well as storing some bile and helping the gallbladder produce bile, or not produce, allowing a pathway for the gallbladder, which produces some bile to aid in digestion. So again, this is a very accessory organ. It's not right on the pathway, but it's heavily involved, right? It monitors glucose, which we break down from starches, and then also is a huge contributor to bile, which allows us to break down lipids. And so then we have the gallbladder, which is attached to the surface of the liver, and it stores excess bile produced by the liver. So we'll look at these 3 accessory organs in a little bit more detail here. So we can see this is the liver here, this big chunk, super important again in like cleansing our blood, in monitoring glucose levels in our blood, and creating bile, which we need to emulsify fats. So here is the liver. This is the little gallbladder. It's just like on the surface of the liver right here. And then, so this is storing any extra bile so that it can just go directly into the duodenum. And then here we have the pancreas. So the pancreas, again, there's pancreatic juices come out of here, flow into the duodenum, so that, again, that produces the pancreatic amylase, trypsin, and lipase so that we can break further down our enzymes. So we're going to look quickly at the enzyme conditions. So we learned a lot about different enzymes and what they need to work. Sodium bicarbonate, for example, was needed to neutralize the chyme so that the pancreatic enzymes would work. So they wouldn't work if it was superacidic conditions. So there's kind of two big things that we are going to look at in terms of conditions that would affect enzymes in the body. So the internal body temperature. Digestive enzymes work best at a normal human body temperature. So if your body temperature suddenly dropped or you got a big spiking fever, your enzymes aren't going to work as well. They work the best under typical human, whatever your normal body temperature is, so about 36, 37 degrees Celsius. And then really, really important, what we just talked about with sodium bicarbonate is pH levels. So each part of the body has different pH levels, and so enzymes work best at the specific level of acidity or basicness that that enzyme is in that area of. So in your mouth, it's obviously a very different condition than in your stomach or in your small intestine. So salivary amylase works best in the conditions that are created with your saliva and the temperature of your mouth. And then moving on, the stomach, very, very acidic. So the digestive enzymes in the stomach work the best under that very acidic condition. We need that sodium bicarbonate in the duodenum to allow those pancreatic enzymes to work better in that less acidic condition. So pH levels are super, super important. And there's actually experiments that if you put, say, salivary amylase into very acidic conditions like the stomach in a lab, the salivary amylase actually wouldn't work and the digestion of starch would not begin because those salivary amylase enzymes aren't used to working in those conditions. But that's all we're going to talk about for the digestive system. Like I said, we're going to look at a virtual dissection next class. If there's any questions on any of this, I hope that you've been updating your enzyme sheet so that you can keep track of all the digestive enzymes and what they do. Like I said, look forward to a virtual dissection next class.